Assalamualaikum and hi everyone. We are from M3822638 and our group will be presenting about genetic engineering in rice. Plant drought tolerance is a complicated quantitative trait that has a complicated phenotype and affects various stages of development. The primary goals of this review were to provide a comprehensive overview of the information on drought resistant genes, QTL analysis, gene transformation and marker-assisted selection, as well as to highlight the current method of developing a durable drought-resistant rice variety through conventional breeding and the use of biotechnology tools. Alright, let me explain a bit about its background. Over the past few decades, drought research has become increasingly important due to its increasing frequency and significance for crop productions. However, it has been difficult to study drought responses due to the drought-tolerant traits, quantitative and complex nature. In addition, crop cultivar subspecies and species have all demonstrated genetic variation in response to drought tolerance in the same environmental conditions. This demonstrates the significance of diversity as a major factor in drought tolerance and in studies relating to drought. Drought-related research frequently focuses on cultivar with high drought tolerance, which are the most promising sources of drought-related genes for future crop varieties.
For case evaluation, we have two methods. First method is marker-assisted pedigree selection (MAPS), have an ability to combine a large number of genes that control both biotic and abiotic factors. Pedigree selection has major drawback of taking a lot of time and necessitating the evaluation of numerous lines on a regular basis throughout the planting season. Pedigree selection is one of the breeding methods that require a high level of familiarity with the breeding material. Second, marker-assisted recurrent selection (MARS) involves making multiple crosses to acquire advantageous allele while preserving genetic diversity. It creates breeding lines with a range of genetic, more precise genetic gain, and shorter, clearly defined breeding cycle. This method has been used to improve grain yield, kernel weight, and percentage of protein in the grain in wheat with success.
Next is proposed solution. We have proposed two solutions which is MABC and GS. The first one is MABC. MABC to select for target loci by reduce the length of the donor segment that contains a target locus and speed up the recovery of the RP genome. It also used to develop near isogenic lines by minimizing carryover donor segments flanking the target locus by providing precise integration of individual genes for detailed characterization of the QTL. The second one is GS. GS can predict the genetic values of selected individuals which depend on genome estimated breeding values, which is GEBV, by using high density markers that are distributed throughout the genome. The GEBV prediction model combines genotypic data with phenotypic data with their pedigree and increases the prediction accuracy.
As conclusion, there are three main advantages to using MAS for crossing. First, selecting a linked marker to an allele from donor cultivar and a locus closer to target gene interest can improve the accuracy and effectiveness of selection for difficult phenotypic traits. Second, it permits the integration of a desirable gene from donor parent to recipient parent while preserving the recipient parent essential characteristic. Third, conventional breeding method necessitate more swapping in each back cross generation in order to transfer a recessive gene, resulting in practice that is too low most breeding purpose. For recommendation, we have decided that the marker-assisted backcrossing (MABC) is the most effective way to produce high quality of drought tolerant in rice. MABC is more popular and effective than the marker-assisted pedigree selection (MAPS). We are not choosing MAPS as our problem solution is because the method takes a long time, require predict evaluation of numerous lines throughout the planting season, and require keeping a record of selection of a major criterion.
The first one is foreground selection. Breeders select plants having the marker allele of the donor parent at the target locus. Then, the selected plants are self-pollinated and progeny plants identified that are homozygous for the donor allele. It can also be used to select for reproductive stage traits in the seedling stage, allowing the best plants to be identified for backcrossing. The second step is recombinant selection. Recombinant selection is to reduce the size of the donor chromosome segment containing the target locus. It also can minimize the size of the donor chromosome segment, thus reducing linkage drag, which called a universal enemy of the plant breeder. The last step is background selection. Background selection is to reduce unnecessary genes by using molecular markers. It also is to accelerate the development of NRP with an additional one or more genes has been referred to as variety development or enhancement and complete line conversion.